Good afternoon, everyone. This is Nelly Poyak from Dana Farber, and this week we again have the Connect Science seminar series. Thank you for everyone for coming and attending these seminars. As a reminder, the talks will be uploaded at the website that's on this first slide, the DFACC website, so you can look at it at a later time in case you don't have time to listen today. And also as a reminder, we have Q&A at the end of the seminar. You can submit questions in the Zoom function. There's a Q&A button there. Please don't send directly to the panel. It's better to have the Q&A function and submit questions there, and the speaker will be able to answer as many questions on live as we can today, and then has access to the questions later on so he can get back to you as well. So it's my pleasure to introduce today's speaker, Todd Golub, who is the founding director of the cancer program at the Broad Institute of Harvard MIT. He's also a professor of pediatrics at Harvard Medical School and the Charles A. Dana investigator at Dana-Farber Cancer Institute. And today, Todd will talk about the phenotypic screening approaches to oncology therapeutic discovery. Todd, thank you for doing this.
Thanks, Nelly, for having me, and thanks everyone for joining in the discussion today. I'm excited to tell you a bit about some of the work that we've both recently published and some unpublished work as well. Just brief disclosures. I'm an advisor to a number of companies and have some industry-sponsored support, but will not be discussing any of those projects today. So what I'm going to talk about today really, I think, is best thought of in the context of cancer precision medicine, which really doesn't require any introduction, I'm sure, to this audience. The idea that you would have some molecular characterization of a tumor, whether it's the genome or other aspects of the tumor, that would then inform the selection of targeted therapy that would hopefully result in a durable response for the patient. And of course, this works occasionally in the clinic, but I think it's fair to say that for most cancer patients, this doesn't yet fully work. And I think one challenge that we need to address is that of where will the targeted therapies come from? This cancer precision medicine rubric assumes that we have a complete pharmacopoeia of drugs that would attack all useful vulnerabilities in tumors, and of course we're far short of that. And so much of what I'm going to talk about today will be some thoughts around how to think about where those new therapeutics might come from. At a very simplistic level, one can divide the world of cancer therapeutics discovery or any therapeutics discovery into two basic categories: target-based discovery and cell-based discovery. And of course, target-based discovery, where you have prior knowledge of the target you want to go after, is the foundation for drug discovery and should continue to be the foundation for oncology drug discovery. Its limitation is that you need to know the target in advance. And more than even just knowing the target, you need to know the desired mechanism of action of a small molecule attacking that target. And I think this is in part because of approaches to screening that we've had in biochemical assays led to a bit of a bias in terms of screening for inhibitors of protein function, but leaving on the table more novel mechanisms of action that be beyond inhibitors of enzymatic activity, for example. We also know that it can be challenging to go from a compound that has activity in a biochemical assay to having that have a similar function in living cells or organisms. On the other hand, there's cell-based screening, which is attractive because you're screening for activity in a living cell. But limitations there are that you're limited by findings that are relevant for the particular model that you're screening. And in the case of oncology, this is particularly challenging because we now know that there's enormous genetic and epigenetic diversity of cancer. And so that presents an inherent limitation of screening, for example, a cell line or a cancer model for a particular activity, because that may not generalize. In addition, many cell-based screening efforts have gone into a kind of permanent black hole because of the difficulty in getting from a small molecule activity to enough detailed mechanistic understanding of that compound's action to be able to optimize it and make it a real drug. That is changing, I think, with the advent of functional genomics and CRISPR-based approaches, and I'll talk about that a bit. This challenge of how do you get beyond the limitation of a particular cancer model on a screen, though, is the topic of the first story I'd like to tell, based on a method we're calling PRISM. This was initially developed by Channing Yu when he was a postdoc in the lab. And the concept here is that it'd be nice if you could screen a library of small molecules for their anti-cancer activity across not just a cell line, but a large number, hundreds of cancer cell lines that represent the genetic diversity of human cancer. And to do that efficiently, we developed a barcoding method whereby we insert a random barcode randomly into the genome of human cancer cell lines. We've now barcoded more than 800 of such lines. That barcoding then allows you to pool the lines together, and in a single well of a multi-well plate, test the activity of a compound against those pooled cell lines, whereby the viability of a particular cell line is inferred based on the abundance of the barcode sequence. I'm not going to go into this technical detail because it's now been published, but we've shown as a very robust method. So if we take it for the purpose of this discussion that there's a method in place to screen large numbers of compounds across large numbers of human cancer cell lines, what might you do with such a capability? Here, Stephen Corsello, a physician scientist, postdoctoral fellow in the lab, said, well, a great first place to look might be with existing drugs. And so Stephen undertook an effort to assemble a large collection of FDA-approved drugs, drugs in clinical development, and compounds that have well-established mechanism of action, even if they may not be in humans quite yet. It turns out that this was much harder than expected, not only to acquire samples of these physical compounds, but even to have a comprehensive list, an annotated list of what all these compounds were. So Stephen did this and reported this in a paper in Nature Medicine now a couple of years ago, and importantly took the step of validating the identity and purity of each of these now more than 6,000 compounds in the library. And at least I was shocked to see that when he did that, and these compounds were obtained from reputable chemical vendors, more than 75 vendors, 28% of all the compounds failed quality control measures, either based on the purity of the compound or the identity of the compound. The chemists online probably aren't going to be shocked by that number because that's sort of expected. I think biologists who aren't used to working with small molecule compounds probably see this as a surprise, but we should take it as a general warning that the identity and purity of compounds should not just be assumed based on the vendor, but should be tested empirically. But so with a cleaned up library of validated compounds in hand, Stephen could ask some interesting questions, such as if you put those through and this is the first 4,500 compounds from the repurposing collection, through this PRISM platform of 578 human cancer cell lines, what do you see in terms of anti-cancer activity? This is a bit of a complex slide. I won't go through it in detail, except to see that you clearly see enrichment for known chemotherapeutic drugs. That's not a shock that chemotherapies kill cancer cell lines. It's also not that much of a shock that you see evidence of killing by targeted compounds, kinase inhibitors and other non-classic chemotherapeutic compounds, killing at least a cell line across this panel of 578. But what was particularly interesting is that about half of the compounds from this repurposing collection that showed some evidence of killing, at least at initial high screening concentration, were non-oncology compounds. That is, compounds that had been developed for non-oncology clinical indications. And so those were deserving of further follow-up screening in dose response, because of course, at a high enough concentration, probably any compound can kill something. And so doing this in appropriate dose response is important. One way to look at the results of such a screening effort is shown in this dimensionality reduction plot called a UMAP, which is similar to a TSNE or principal component analysis, where we're looking at the classes of compounds that are shown here and color-coded, not based on their structural similarity, but rather based on their similarity in killing profile across the 578 cell lines. And so what you can see of interest, for example, towards the top of the screen, I don't know if you can see my mouse, all the MEK inhibitors cluster together, not that far away from RAF inhibitors. That kind of makes sense, makes you feel good. The EGFR inhibitors, in orange, largely clustered together. Tubulin stabilizers or destabilizing agents all cluster in a similar part of the map. What's interesting, though, is that you also see clustering of compounds with similar killing profiles across the 578 cancer cell lines that you might not expect. For example, all of the statin cluster together, HMG, CA-CoA reductase inhibitors cluster together. Glucocorticoid receptor agonists, which we tend not to think of as strong anti-cancer compounds, all cluster together. In fact, they're not strong cancer killers, but because you're looking across such a large number of cell lines, even minimally growth inhibitory compounds that have a consistent pattern of killing across the large panel of cell lines behave as a coherent unit. And so even just as a biological discovery mechanism, this large panel of cell lines by compounds is proving interesting. Now, perhaps a more useful way to visualize the data with respect to compounds worthy of follow-up is shown here. Separating the compounds that are either known oncology agents or non-oncology drugs on the right. And the two axes that we're looking at here are on the x-axis, what we're calling a bimodality coefficient. That is a statistical measure of the extent to which any given compound results in a clear distinction of either killing a subset of cell lines and not killing others, as opposed to a broad gradient of killing most cell lines in a concentration-dependent manner. Broadly, cytotoxic compounds would not have a bimodality coefficient, whereas compounds, for example, EGFR inhibitors that largely kill EGFR mutant cell lines have a high bimodality coefficient, and those indeed score highly in the known oncology space in red. The y-axis is a measure of the predictability of such killing based on the baseline molecular features of these cell lines, which have been all extensively genomically characterized as part of the Cancer Cell Line Encyclopedia project. Genome sequencing, RNA expression, metabolite profiling, and so on. And so the most interesting compounds are those that are in the upper right-hand quadrant, those that have a bimodal pattern of killing, and that pattern of killing is highly predictable based on the molecular features. And while they're not labeled here, all these data are now publicly available. In red, in the upper right-hand quadrant, are examples of the known targeted agents, targeted kinase inhibitors, for example. But what's of greater interest, I think, for the purpose of this discussion is what's in blue in the upper right-hand quadrant, indicating that there are indeed exist non-oncology compounds that selectively kill some cancer cell lines, but not others, and that killing ability is predictable based on genomic features. I'm going to show one of those examples on the next slide. And this, again, is all the work of Stephen Corsello. And just to give you a sense of an example of this, and this relates to a predictive biomarker of ABCB1 expression. As you know, ABCB1 is a component of the multi-drug resistance protein, MDR, also known as P-glycoprotein. At the top, you see that there's a gradient across the 578 columns illustrated here. There's a gradient of ABCB1 expression from high-level expression in red to low level of expression shown in white. And shown below that is a heat map looking at drug sensitivity, where those drugs, which are different rows, if they kill cell lines, the killing is shown in blue, whereas those cell lines that are resistant to killing are shown in white. And here you see a very clear correlation between high level of ABCB1 MDR expression and resistance to these known agents. For example, on the bottom, paclitaxel, well known to be resistant when MDR1 is highly expressed. This makes you feel good about the quality of the data set, but of course, this isn't anything new. What was interesting, however, is the following, which is that there was a single drug called tepoxylin, which had the opposite pattern of killing. That is, tepoxylin only kills those compounds that are MDR high and spares those cell lines that do not express ABCB1 or MDR1. What's tepoxylin? I hadn't heard about it before. It goes by the trade name Zubrin. It turns out that it's been FDA approved for use in doggies and was developed as a dual cyclooxygenase 5-lipoxygenase inhibitor, which at least we hadn't thought of as being those targets being relevant for cancer or having any connection to MDR1. It also made it to the clinic to phase 2 in humans, also for pain relief and rheumatoid arthritis, but was later abandoned. Importantly, because we had screened thousands of other compounds, we were able to immediately see that there were no other COX or lipoxygenase inhibitors have this cancer-killing effect. And moreover, if you combine a COX inhibitor and a lipoxygenase inhibitor, you can't recapitulate the effect of tepoxylin. So this suggests that the killing by this compound tepoxylin is likely to be an off-target effect. What that target is and whether it has any functional role in connection to ABCB1 is a question, because it could be that ABCB1 is just for some reason, a biomarker, but has no functional connection, mechanistic connection. And so to address this, Stephen performed a genetic modifier screen where he took a cell line that was sensitive to tepoxylin, that is killed by tepoxylin, and then introduced a genome-wide CRISPR-Cas9 knockout library to ask whether knockout of any gene could rescue the cells from killing by tepoxylin. And the result is shown here, where there was a very clear outlier, which was sort of surprising but gratifying, which is that the predictive biomarker of killing itself, ABCB1, was required not only as a predictive biomarker, but is required for the killing of cells by tepoxylin. Stephen did the opposite experiment, which is a CRISPR activation screen, a CRISPR-A screen, and asked overexpression of what genes in the genome could also potentiate the effect of killing by tepoxylin. And a clear outlier there, again, was ABCB1. So again, this suggests, but does not prove, that ABCB1 is actually a direct target of tepoxylin. But of course, the compound can't be killing cells by virtue of its inhibiting ABCB1 function, because ABCB1 genetic loss of function actually rescues doesn't phenocopy the effect of the drug. And so we're still working on the precise mechanism of killing by this compound, but it suggests that it's going to be some gain of function of ABC1 transporter. It's not, we know that it's not simply reverse transport of the compound into cells. We've shown that clearly. It's likely to be some toxic gain of function that was exposed by this cell-based PRISM screen. More to come on this particular compound. Now, the precision medicine, cancer precision medicine rubric needs to take seriously that if you're going to not only kill cells, in vitro, that having durable responses in vivo in patients is going to require a reckoning with the importance of the tumor microenvironment and its role in promoting cell survival, which of course we're missing in its entirety in these purely cell line-based PRISM screens in vitro. And so the next project I'd like to describe is the work of another postdoc in the lab, Xin Jin, said, well, could we take this same kind of conceptual thinking of large-scale pooled PRISM screening and extend it to the in vivo setting in xenotransplanted mice as a way to learn something about the important role of the microenvironment in promoting tumor cell survival in the context of the genetic breadth of cancer that we can capture by looking at large numbers of cell lines as opposed to your favorite workhorse model. And so we don't have time to go into the details of how this works, but the basic concept is that these barcoded human cancer cell lines are pooled, injected into the left ventricle of an immunodeficient mouse so that the cells directly enter the arterial circulation and don't simply get stuck in the lung as they have a tendency to when you do tail vein injections. And then after a few weeks, harvest key organs, in this case, brain, lung, liver, kidney, and bone, and then enumerate barcode abundance in those various organs as a proxy for the ability of these human cancer cell lines to metastasize to different organs. There are different technical approaches to this that involving depleting the mouse cells or not depleting the mouse cells, enumerating the human cell lines by RNA-seq, single-cell RNA-seq, or by just barcode, PCR abundance of the barcodes. I'm going to skip those technical details, but simply to say that these different technical approaches all seem to work remarkably well. I'll just give one example of that as shown here to test the dependence of the experimental format on the findings. Here comparing two approaches to this so-called metastasis map or MET map. One going to the extreme of pooling 500 cell lines into a single pool, injecting all 500 into the heart of a recipient mouse, or doing it more carefully by making only pools of 25 and injecting pools of only 25 cell lines, and then doing that 5 times in 5 different mice to limit the pool size. And on the right, you see the scatter plot of the results comparing, looking at the overall metastatic potential of the all at once 500 cell lines and the more careful only 25 at a time. And you see what I was shocked by is a remarkable concordance of the overall metastatic potential that's pretty much independent of the experimental format, which I think speaks to its overall robustness. Just a moment about doing this in the intracardiac injection approach as opposed to subcutaneous injection. We've done a limited number of these experiments, but Xin showed, for example, that if you inject, this is a pool of 488 cell lines subcutaneously, and then after a couple of weeks of growth, you sack the mice and then look at the rotation of those 488 cell lines. Graft, you see that you can only recover about 50 of the nearly 500 cell lines because presumably because of the local competition for microenvironment and nutrients in a sub-Q injection. What's interesting is that same pool of 488 cell lines, when injected into the arterial circulation, you don't recover them all, but there's evidence of metastasis to at least one organ in about half of those lines, significantly more than the subcutaneous situation. We hypothesize that the reason that you recover more here compared to subcutaneous, or I should say limited experience in mammary fat pad injection with breast cancer cell lines, you see the same as the subcutaneous result, which is that in the disseminated metastasis model, you have spatial separation of the cell lines from each other so that there's not local competition for nutrients and other factors in the metastasis model. So from this, you can generate a map of metastatic potential across all of these cell lines, and I should say that all of these data will be made publicly available through our website for either interaction through the DETMap portal or for download if you're interested in the raw data. But you can get a sense of here looking just at the breast cancer cell lines, that there's an enormous diversity of metastatic potential, ranging from cell lines like MBA231, which is shown in these spider plots. MB231 is a commonly used breast cancer cell line to study metastasis. That shows evidence of broad metastatic potential to all 5 organs. Whereas other cell lines, for example, here, HCC 1395, shows liver selective metastatic potential. And then there are other cell lines shown here that show no metastatic potential whatsoever. So as a resource, we think this will be useful for others to help, for example, decide what cell lines might be most applicable for particular questions of organ-specific microenvironment growth and or metastasis. But can you learn anything biologically from these types of data? And so Shin asked the question, focusing on breast cancer and the ability of certain breast tumors, but not others, to metastasize to brain. And it's known clinically that some breast cancers metastasize to brain and others don't, but little is known about the molecular basis of that. And so here, just showing in this plot, we could use the differential metastatic potential of breast cancer cell lines to brain to ask what distinguishes the highly brain metastatic cell lines from the non-brain metastatic cell lines, and to use the genomic characterization of these cell lines to address that question. Because as I said, we have mutation, copy number, metabolite profiling, RNA-seq, and the in vitro genetic dependency data from the DETMap project to inform and to look for correlates of brain metastatic potential. And the answer here was pretty striking, which is in all of these dimensions, the strongest signal, and I won't go through it in great detail, was one of lipid metabolism. In the case of mutation analysis, we saw enrichment for PIK3CA mutation in brain metastatic breast cancer cell lines. In copy number space, we saw evidence of 8p deletion being associated, at least, again, this is an association, not causality, but associated with brain metabolism, with brain metastasis. Metabolite profiling done with Clary Klisch at the Broad Institute showed that the cell lines with brain metastatic potential had low triglyceride level and high cholesterol levels, and so on. I'll show a little more detail some of these findings, but this was a strong hypothesis. The metabolite profiling of low triglyceride content of those cell lines with high metastatic potential and high cholesterol got our attention because the brain microenvironment, as it turns out, has the lowest triglyceride content of any organ in the body, as shown in the lower panel. This is in normal mouse brain, where, of course, fat adipose tissue has high levels of triglyceride, as you would expect. The brain has very little triglyceride content. And so our hypothesis here is that perhaps it's the case that cancer cell lines that in certain microenvironments can borrow triglyceride from adjacent, for example, adipocytes, as Rich White has shown in the case of melanoma, as a source of lipid for energy metabolism and membrane constituents. Such triglyceride is not available in the brain, and so the only way to survive there is to have another source of lipid metabolism. And so this is why it was particularly interesting that when we looked at the correlations with genetic dependencies, we found that those cancer cell lines that were able to metastasize to brain were particularly dependent on the master transcription factor SREBF1, which is known to be a master regulator of lipid metabolism, suggesting that perhaps these cells are using SREBF1 mediated lipid metabolism as a source of generating critical lipids, fatty acids, as opposed to drawing from high triglyceride microenvironments. Shin followed up that result, now not in the pooled format, but in individual SREBF1 knockout experiments, and showed that while there is some in vitro dependence on SREBF1 for survival, and some evidence in non-brain tissues also for dependence on SREBF1 for survival, that this SREBF1 dependency was most dramatic in the brain, where you saw about a 200-fold loss of viability in the brain compared to wild-type cells, whereas, for example, in the lung, this is only a 10-fold difference in viability with SREBF1 knockout. So more to be followed up on that particular story, and whether modulating SREBF1 or this lipid metabolism pathway has therapeutic potential in cancer, I'm not sure. But I think what we did learn from these in vivo PRISM experiments is that it's now possible to bring the scale of modern genomics and functional genomics to the study of more complex, perhaps more physiologically relevant environments, such as studies of the tumor microenvironment. Now, in the last bit, I want to talk about drug resistance, which of course is also going to be important in this precision medicine framework if we not only want to have initial responses, but durable responses to targeted therapies. Of course, there's much work being done on identifying mutations in drug targets, for example, the T790M mutation in EGFR that explains a lot of EGFR inhibitor resistance in lung cancer. But we've been focusing recently, and the we being Peter Tsvetkov in the lab, on changes in cell state that may not be mutation driven, but changes in cell state that can also confer drug resistance. Peter initially made the observation, published last year in Nature Chemical Biology, that a shift to mitochondrial metabolism can confer resistance to, in this study, proteasome inhibitors such as bortezomib. But it turns out that this shift to a high mitochondrial state has been observed not only in the case of proteasome inhibitors, but as reported by others, a growing number of anti-cancer agents develop resistance when cells move into a high mitochondrial state. So that's interesting and frustrating, but Peter asked the interesting question, well, if there's positive selective pressures for cells to adopt a high mitochondrial state so as to become drug resistant, could one exploit that to look for new vulnerabilities in that high mitochondrial state? And so he undertook a small molecule screen to ask, are there any drugs that are preferentially active in killing when cells are in this high mitochondrial state, which experimentally was done by growing cells not in glucose but in galactose, where you need to adopt oxidative phosphorylation for growth and survival. And so we asked, in the galactose versus glucose state, is there differential sensitivity to drugs? And in parallel, also did a screen, in this case, using proteasome inhibitors as the example, using other mechanisms to induce the proteasome inhibitor state to ask, are there any compounds that preferentially kill those proteasome inhibitor resistant lines? And in both cases, there was a clear outlier, a drug called olesclomol. What is olesclomol? This is a drug that was actually developed by Synta Pharmaceuticals, was brought to clinical trial over a decade ago, without a particular therapeutic hypothesis, and that is not a particular patient, sensitive patient population in mind, and not really with deep understanding of mechanism of action of this compound. It showed some evidence of activity in melanoma in a phase 2 trial, and based on that encouraging result, went to phase 3, where unfortunately it failed due to efficacy. The drug itself is problematic to the extent that it's poorly soluble, and so in the phase 2 trial, they used a sonicator at the bedside to promote solubility prior to injection IV. In the larger phase 3 trial, that was not feasible, and so the drug substance itself is problematic. So it makes it difficult to interpret what to make of a negative trial, particularly in an unselective patient population in with a suboptimal compound. It is interesting that in a post hoc analysis, the company published that they did see some evidence of a clinical signal in those patients with low LDH levels, which interestingly is consistent with Peter's hypothesis of a high mitochondrial state conferring sensitivity to the compound, but clearly not definitive. What can we figure out using modern methods about the mechanism of action of olesclomol? It was known that olesclomol is a copper ionophore, and Peter was able to summarize here, again, recapitulating the results that if you grow the cells in galactose, you see a dramatic left shift, more than 2 orders of magnitude greater sensitivity when cells are in this high mitochondrial state compared to when they're grown in glucose. That if you deplete the media of copper, you can then demonstrate that the compound only kills when you add copper back, as shown in the green line, whereas it does not kill in the absence of copper. And consistent with that, in copper-repleted media, if you treat with a copper chelator, TTM, you completely abrogate the killing of the compound. So copper-dependent killing for sure, but what more can we say about mechanism of cell death? It was suggested in the literature that perhaps olesclomol kills by apoptosis, but here genetically in Baxback knockout cells, it's clear that in contrast to the positive control, paclitaxel, olesclomol kills cells just fine, even in Baxback deficient cells, arguing against this being an apoptotic mechanism. And moreover, when Peter extended this beyond the genetic experiments to small molecule tests, he could show that inhibitors of ferroptosis, for example, ferrostatin, have no effect on killing. Pan-caspase inhibitors do not inhibit killing by the compound. It's only a copper chelator, TTM, which rescues killing by olesclomol. So we take from this that this is a non-apoptotic, non-necroptotic, non-ferroptotic mechanism of cell death that is copper dependent. And for that reason, we're calling this mechanism of cell death cuproptosis. We'll see whether you all like that name and it becomes sticky or not. This still doesn't tell you what the target of alescomol is. And so for that, Peter again turned to genome-wide CRISPR knockout studies to ask knockout of what gene in the genome could rescue killing by alescomol. And here he found that there was a strong outlier, the reductase FDX1, which when knocked out completely conferred resistance to alescomol. He also showed on the right-hand panel, looking at the PRISM profile of alescomol, that the top predictive biomarker of killing by alescomol was again FDX1. So top score in a biomarker analysis, top score in a genome-wide CRISPR modifier screen points to FDX1 as being essential for killing by this compound alescomol, which, as far as we're aware, no one had previously considered. In collaboration with John Markley at the University of Wisconsin, we were able to show, based on NMR, that alescomol indeed is a direct binder to alescomol, a recombinant, a direct binding recombinant FDX1 protein. And we're able to show that alescomol does, at least at high concentrations, have FDX1 reductase inhibitory activity, but that FDX1 inhibitory activity cannot explain killing because, again, as I showed you with the ABCB1 PRISM example, knockout of FDX1 rescues compound killing by alescomol. It doesn't phenocopy it. So loss of FDX1 function cannot explain killing. It may have this activity, but doesn't explain it. To further try to figure out the mechanism of action of this compound, Peter did another CRISPR knockout screen here, focusing on 3,000 metabolic enzymes, looking at the intersection of two screens, one with alescomol itself, and the other with another copper ionophore, disulfiram, FDA approved for alcohol abuse, that shares the killing pattern of alescomol across the PRISM profile. And in addition to recovering FDX1 as being required, as I just showed you, required for killing, he also found all these other genes, essentially none of which I'd ever heard of, but all of which are part of the lipoic acid pathway, which I confess I hadn't really thought of a lot either, but that's what happens when you do these unbiased screens. I also mentioned that this limited number of 10 genes that came out of this modifier screen was also MTF1, a master transcriptional regulator of metallothioneins, which are metal binders and copper binders included. So that makes sense, that if you modulate the abundance of a metal chelator in a cell, that would modulate the sensitivity to a copper-dependent killing mechanism. In experiments I don't have time to go into, Peter has also showed that FDX1 is actually a regulator of the lipoic acid pathway, and here shown by immunohistochemistry done by Sandro Santagata at the Brigham, could show that wild-type cells express high levels of lipoic acid, but that if you knock out FDX1, you lose lipoic acid expression, showing that FDX1 is upstream of this entire pathway. What the hell is this pathway? It turns out that, remarkably, that lipoic acid is a metabolite, a covalent metabolite modification of proteins on particular lysine residues in a particular context. There are not a large number of proteins that are lipoylated, that is modified by lipoic acid. It's a handful of enzymes, and they're all part of the TCA cycle and related to mitochondrial metabolism, which of course is interesting because now we've established the connection between lipoic acid and alescomol and the high mitochondrial state that I started this part of the talk with. So a number of the enzymes that are involved in this are, for example, pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase, are all activated by lipoic acid modification. But what does copper have to do with any of this? How do you connect copper dependence to this lipoic acid metabolite mechanism? It turns out, shown not by us but by others, that lipoic acid itself is a super high affinity binder of copper. And so our working hypothesis, not yet fully established, is that this high mitochondrial state requires FDX1 reductase activity for function. We know that FDX1 knockout cells cannot survive in a high mitochondrial, can't adopt a high mitochondrial state without FDX1. That the FDX1 reductase reduces copper 2+ to copper 1+, which is known to be more toxic to cells, and that the model suggests that this toxic copper is bound by lipoic acid modified metabolic enzymes in this pathway that either create toxic intermediates of these enzymes specifically, or result in the accumulation of toxic copper in the mitochondria, resulting in a nonspecific copper toxicity, which, as you know, copper is normally very, very highly regulated within cells. And typically the intracellular concentration of copper is almost non-existent under normal conditions. It's highly regulated by chaperone proteins. So this is the working model, still further work to be done. So let me conclude with just touching on these 3 vignettes that I described. First, on PRISM as a strategy to allow for the discovery of compounds with, in particular, what I'm most excited about is novel mechanisms of action. If you're looking for an inhibitor of a known enzyme, for example, PRISM's not a particularly smart way to go about it. There are better ways to do that. But it's looking like a surprisingly large number of the novel activities of even existing drugs and non-oncology compounds look like they're having novel gain-of-function activities. For example, stabilizing protein-protein interactions, as an example. Those types of things that are hard to discover other than in a cell-based assay. This in vivo or METMAP, metastasis map project, I think, gives an example of the feasibility of bringing large-scale tumor microenvironment studies to the lab, which is interesting. Still not a substitute for the detailed mechanistic follow-up of individual models and perturbations, but makes it possible to do larger scale studies in perhaps more physiologically relevant settings. And then the last story, highlighting the importance of mitochondrial metabolism in drug resistance, but pointing out that that problem also creates a novel vulnerability, and that is to cuproptosis, copper-dependent cell death that can be induced by alesclimol and other copper ionoforms via this, what we think is a novel mechanism of lipoic acid modification of key metabolic enzymes. With that, I'm happy to open it to questions. I want to thank the people that have led this work, many more collaborators, but in particular, PRISM work led by Stephen Corsello with respect to discovering new indications for non-oncology drugs. Xin Jin led the metastasis map project, and Peter Tsvetkov the cuproptosis project. And with that, I'm happy to take any questions, and I guess turn to Nellie to moderate if there are any questions.
Yes, thanks, Todd, for a great talk. And there are a number of questions. I tried to group them according to the areas you covered, and maybe also combine a couple of questions into one. So there are a number of questions related to more like somewhat technical details of the PRISM screen, doing these large scale screens, specifically like, do you include normal cell lines, and do you consider other measurements besides viability for such a screen in vitro? And some people were wondering the specifics of how can you maintain 500 cell lines at once. I guess that's more technical, but anyway, if you could comment on some of these, like the what is the, how would the doubling time affect the screen? I think you probably controlled all for that.
Yeah. So yeah, I'm happy to speak to, those are all great questions. First, we don't keep pools of 500 cell lines growing in continuous culture for long periods of time because you can't maintain the abundance of particular cell lines in the pool if you do that. The pools are generally made in pools of 25, in subpools of 25, and then those subpools pooled together at the time of an experiment. And then discarded after a particular screen rather than kept in continuous culture. Another question is, well, don't you get cell-cell interactions and paracrine effects of one cell line and the other to another, and can't that screw up drug sensitivity findings? And that's theoretically possible, and my whole microenvironment part of the talk would suggest that yeah, those kinds of factors can be important. So far, interestingly, we haven't seen evidence of that in the PRISM format, and it could be that because any one cell line is only represented in a minority of a large pool, that you don't have the potential for that paracrine effect to be seen, and it's that those signals are simply diluted out. So we haven't seen evidence of that, even though it's theoretically possible. I think I'm forgetting, Nellie, one other aspect of a technical
the doubling time and including normal cells, including immune cells in a screen.
Yeah. So doubling time, we do pool based on an approximation of doubling time to pool the fast growing cell lines together and the slower ones in separate pools. We've discussed normal cell line components of the pool, but haven't yet done that. What a normal cell line actually is probably a matter of debate. I think it probably is a good idea. Some people would say, well, would you be prepared to really throw out a candidate because it also killed a cell line that was technically normal, but still is growing, proliferating in culture? I think that's a matter of philosophy, but I actually think it is a good idea to try to do that. We've thought about doing this in heterologous cell culture with immune cells. Haven't thought about doing that yet in the context of a PRISM pool, which would be really complicated with multiple genetic backgrounds and so on, but that's an interesting idea.
Mm-hmm. Related to the screen is like, are these barcoded cell lines available to outside people?
Yeah, that's a tricky question. We've had to be compliant with ATCC and other repositories, which restrict our ability to make cell lines available. So at the moment, we're not able to make the whole PRISM collection available to the outside, but we are open to collaborations for people that want to screen their compounds using the PRISM format.
Maybe the library to make the barcoded cells could be available.
Absolutely, yeah. Very happy to do that.
All right, so there are a number of questions about the metastasis screen. Specifically, how do you think the microenvironmental interactions may influence the metastatic potential? Why so many breast cancer cell lines grew in the liver? And also, do you see within each cell line a particular subclone growing to certain sites?
Yeah, so I don't think we've looked at the subclone question in great detail, but that's an interesting question, whether there is selection for a minor subclone that has metastatic potential. There is evidence from the work of Joan Massaguet's lab and others that if you take cell lines in a xenograft setting, find a metastasis, pick the metastasis, and then reintroduce into a metastasis model, that the metastatic potential goes up, suggesting that there is some selection for high metastatic potential. The question of why the liver? I think one issue is that the liver is big, and we haven't attempted to correct for organ size as a function here. But I think it is pretty clear that the different tumor environments do have different survival factors that are supporting growth in one organ versus another. Now, it could be that there's a seeding problem, and that if a given cell line could seed a particular organ, it could survive there just fine. Shin has done some experiments in that case in the breast cancer model, for example, with SREBF1, where if he directly injects cells into the brain, thereby abrogating the seeding question, and he's been able to show that they still have an SREBF1 survival defect, suggesting that there is that the tumor microenvironment per se isn't important for survival in that particular microenvironment. Okay.
So there are a number of questions on a coprotosis. Looks like people like the name already. For example, what is the physiologic, is there any physiologic equivalent for coprotosis induced, some endogenous trigger that would induce that? And do you see any particular, morphological changes of this type of cell death?
Yeah, that's a really interesting question. I'm not aware of unlike apoptosis, which of course has an important physiologic role in development and other settings. I'm not aware of a copper-mediated cell death mechanism that's normal physiologic response, or rather is that this simply a mechanism that's being co-opted by drugs? I would guess that it's probably the latter. And that in normal physiology, there's not a role, but we haven't proven that explicitly.
Yeah, one of the attendees suggested maybe in Wilson disease. Do you see this coprotosis? That's the metabolic disorder with the high copper accumulation.
Yeah, we haven't looked at that. There's a known cancer association with Wilson's disease, whether that goes in the right direction, or not in terms of what you'd expect in terms of copper-dependent cell death. I don't know. There's an interesting literature that you actually need cell death in order for tumors to develop through cell competition. So I guess you could do some hand waving to imagine how increased cell death in Wilson's disease with copper overload allows for tumorigenic clones to take over, but that's pure speculation.
Okay, and then a couple of questions. Well, actually, we have time. Maybe one more question about the FDX1 is how do you explain the FDX1 role and the interaction with LS-clomol compound, like the formation and the lipoic acid? So there are a number of questions on this. What does it suggest? Right.
So we're still trying to figure this out. We know that you need FDX1 for expression of the lipoic acid pathway. So without FDX1, you don't get expression of these enzymes involved in lipoic acid modification, and then you don't get lipoic acid modification of those proteins. But in addition to that, we also have evidence that FDX1 is the direct target of LS-clomol, and so the running hypothesis is that in addition to its regulatory role for the lipoic acid pathway, the reductase activity of FDX1 reduces copper 2+ to a more toxic copper 1+ state, and also directs it to the mitochondria where FDX1 is particularly expressed. That's the current model, but still a lot to be worked out.
Okay, maybe that's one question related to the mitochondria. Like, do you see changes in ROS and types of ROS during coprotosis? Good link to the mitochondria.
Yeah, we haven't looked explicitly at what's happening during this process of cell death that deserves more attention. Haven't done that yet.
Okay, thank you. That's all we had time for today. And Todd, thanks again for the great talk, really stimulating. And I'm sure a lot of people will go to the website to look up the data. And as a reminder again, there is, the slides will be uploaded on the website, the DFHCC website. And all the questions that we haven't been able to answer live, Todd will have access to them and he can get back to you later on. So thank you for your attention. Thanks for attending. Thanks, Todd. Bye.
Thanks, everyone.